Hi, my name's Allie Bouchard, and today I'm going to be discussing how environmentally induced nutritional limitations disrupt polyethism in our Apis mellifera colonies. So, polyethism, what is it? I'd never heard the term before taking this class, but it's actually a task differentiation superpower, in my opinion, amongst honeybees, where tasks are done according to any demands that the colonies may be experiencing at the time, as well as the bees' ages. So this coordination matters for efficiency, flexibility, and overall survival of the colonies. But I want to highlight flexibility because bees can actually shift roles if the colony is experiencing some type of stress or pressure. So we will dive into that a little deeper. So, do these disruptions actually occur? Should be the main question. And if you're asking that question, you're already on the right track because while Apis mellifera is highly organized as a species, environmental pressures are still incredibly concerning. So, in my research, I wanted to ask how experimentally induced pollen limitation alters juvenile hormone and vitalogenin levels, as well as the overall colony health and structure. Where pollen, a major nutrient, is for some reason scarce. It could be pesticide use killing off our plants or something of that nature. And vitalogenin, a storage protein within the bee, is an indicator of that scarcity. For example, if you're intaking high amounts of pollen, we'd expect to see a high corresponding level of VG or vitalogenin. So now that we've established honeybees experience this division of labor, we know that they do so primarily on the basis of age. But how do we know this and what is the proper division? So our younger bees are called nurse bees. They're responsible for taking care of the developing brood, and they're more physiologically apt to doing so with well-developed hypopharyngeal glands and a low juvenile hormone level. And they are, like I said, directly responsible for brood survival. Forager bees, on the other hand, are older bees, and they're responsible for resource acquisition. So they collect nectar, pollen, water, and they communicate via the waggle dance, which lets other bees know where a food source would be. And they are more physiologically prepared for their position because they have reduced brood food glands, so they wouldn't be good as nurse bees anymore, a high juvenile hormone level, and an increased metabolic capacity for flight. And they directly determine colony growth and survival. So on the right here, you'll see figure A is met and B is KR-H1. And these stand for methoprene tolerant and Kruppel homolog 1. So these are cells that respond to juvenile hormone or a key gene in the KRH1 respect, which is activated by juvenile hormone. So these are markers of levels of juvenile hormone, and we see that they're much lower in nurse bees than foragers. So now that we've established a difference, should we even be concerned about certain environmental changes impacting such a seamless process? And the answer is yes. So in this study, for example, that I've cited here, researchers basically wanted to see if, environmental pressures of sorts would impact polyethism or elicit a certain change in the procedure, which they end up finding to be true. So when they removed foragers from colonies, the colony compensates by nurse bees transitioning earlier into foragers or early onset foraging. And when they kept foragers inside or contained to modulate severe weather, for example, nurses delayed foraging, so delayed onset foraging. Thus, we know that what goes on around the bees indeed has a significant potential to impact the functioning of their task differentiation mechanisms. So if the environment can impact Apis mellifera polyethism, what specifically should we be concerned about? And in 2026, like I said, deforestation, pesticide use, things like that, anything that decreases our pollen availability comes to mind. So for bees, nectar and pollen are both nutritionally significant. Nectar serves as a carbohydrate source, while pollen satisfies their protein, lipid, sterile, micronutrient requirements. Thus, I would say pollen is our key player in sustaining our bees. And in our investigation today, we will examine vitalogenin, or VG, which is a major indicator of bee immunity and longevity. So VG is synthesized primarily in the fat body of the abdomen, released into the hemolymph, and incorporated into those hypopharyngeal glands that we referenced earlier, which produce royal jelly proteins to feed the brood. And we can use VG as a marker of pollen being received. And this is just a cool AlphaFold model of phytolacinin that I provided on the right. So from my research, VG can give us great insight into the behavioral maturation, polyethism, normalcy, and the demographics, if you will, of our colonies. And with our figures on the right, we see that a low pollen consumption decreases our phytolacinin, which ultimately leads to collapse. High brood feeding in conjunction with low phytolacinin, acting as regulator, also leads to collapse. So VG is essential for regulating division of labor and preventing colony collapse. But, everything does come at a cost. So when we restrict pollen, we reduce phytolacinin levels in nurse bees, forcing them to reallocate the resources toward the queen because, they're trying to sustain the population of their colony first and foremost. So this imbalance accelerates behavioral transitions from nursing to foraging and thereby disrupting normal age polyethism. So these figures on the right here, the black region would be survival of the colony, whereas the white region is extinction. So we see with low pollen collection, it leads to starvation and VG depletion. So pollen scarcity shifts resources to the queen, depleting phytolacinin and destabilizing the colony. So we've covered now that, Apis mellifera's highly advanced polyethism, as well as the effects of environmental triggers and the markers thereof. And we see that phytolacinin and juvenile hormone levels are both impacted by environmental triggers and nutritional limitations. And in this case of pollen restriction, early foraging commences in hopes of finding additional resources for the colony. Excuse me. But why should we care? After all, they're just bees. Well, pollination is responsible for our food security, and that's no secret. I think the actual figure is a third of our global crops, but in any case, any disruption to Apis mellifera would threaten our availability of certain crops. They also support ecosystem stability, not just other species, but including humans, and the advancements of a personal passion of mine, which is medicine, which we often get from plants. And essentially, any reduced pollination would decrease our crop yield, which leads to higher food prices overall. So in this takeaway, we would know that small changes in nutrition can cascade into colony level failures, emphasizing the need for better environmental and agricultural practices. And here is our bibliography. Thank you.